Man, that burns, that hurts, right? Right away, I need to make clotting protein. Wait a minute, clotting protein? That's a protein. How was protein made? From translation, at a ribosome. Where does it get the instructions? From mRNA, which comes from transcription. See where this is going? So my body says, whoa, 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 bad, bad, we got to cut. We got to cut here. Yikes, we got to cut. So then my body says, all right, let's go. Go to the gene. Where's the gene at for clotting protein? Right there. Go. Transcribe it, transcribe it, transcribe it, translate it, translate it, translate it. Make a whole bunch of clotting protein so it can float around in my body, go to the paper cut, and prevent me from bleeding out, right? And you're saying a paper cut? It's that complicated? Yes, it's that complicated. Isn't it amazing your body does it just like that? It is amazing. So that's prokaryotic. We're going to move over to eukaryotic side, okay? So if you don't want to hear the eukaryotic gene regulation, you can leave now. I wonder if there's anybody over there that wants to hear about. Can you imagine not coming to this workshop and understanding this?
Hey, anybody over there?
We're moving on to eukaryotic. And I hope those two boys in the corner are doing a nice job of being on task. Right. Okay, so it gets a little crazy when it comes to, I talked about that already. Let's move on. Is there any questions over prokaryotic, or did I explain that pretty well?
So what happens when you're lactose intolerant?
What happens when you're lactose intolerant? That means you do not have the enzyme. We are like E. coli in that sense. You do not have the enzyme to break down lactose. So your body kind of treats it almost as a poison almost and says, what do we do with this? Because somebody who has the enzyme breaks it down, but somebody who's intolerant is intolerant. They can't break it down. So, but everybody knows I can break it down. Okay. Yikes, tough crowd here. Anyway. That's rough. It's funnier in real life than it is. Okay, well, anyway. So, let's reset our brains because it's totally different with eukaryotic cells. It's totally different with eukaryotic cells. Eukaryotic gene regulation simply says, there's not, what here is the same? RNA polymerase, right? I mean, there's still genes being transcribed. That's true, but we kind of already knew that. Well, what would serve as the promoter? We had a promoter in prokaryote. What's the promoter in eukaryote?
The transcription factor.
What do you think?
The transcription factor.
Nice shot. You can call it the TAT box, or sometimes we just call it the TATA box. That's what it was called. And it's not really, basically what the TATA box is a bunch of, it might be something like that. It's not like just TA, okay? It's basically a bunch of DNA. This is DNA right here. And it is a bunch of T's and a bunch of A's, and are in that order. It could be, 2 A's, 2 T's, or whatever. Remember what A and T stand for? Adenine and thymine. That's all it is. It's nothing new here. We're not like throwing some new thing at you. You already know about thymine and adenine, okay? So, there's this thing called an enhancer, and that's where the transcription... Wait a minute, what's a transcription factor? Well, a transcription factor does a whole bunch of stuff. What are some things a transcription factor does? It helps with transcription, right? Yeah, okay. Let's look at some list of things here it goes through. So, transcription factors, the roles of transcription factors are they help the chromosome kind of unwind and unpack. They attract the RNA polymerase. You can kind of see that a little bit. Actually, this is kind of cool what it does. Some of the transcription factors hit the TATA box. Some of them hit the enhancer. And what it does is it kind of curls up. You see that? Why do you think that would be an advantage? Well, does the RNA polymerase know exactly where to land in that situation? Yes. I would think so. I mean, it's kind of curled up almost. It kind of is like a cup. And somebody described it 4 or 5 like a baseball mitt, right? In a baseball mitt or a softball mitt, you kind of have that part that curls around. You can catch it. Ah, yeah, okay. So, anyway, the transcription factors also block access to some genes. So it can turn on genes and it can turn off genes. And that's the whole point of this whole chapter. Most of the time, the transcription factors are going to be really important in saying, hey, RNA polymerase, come here. This is where you belong. This is where I need you. And this is where we need to start transcribing. He's bleeding, man. We need to make some clotting protein. So here's the gene right here for clotting protein. It zips along there and it makes some what?
Copies.
What does it make?
Enzyme. Protein. Amino acid.
mRNA.
mRNA.
Come on, folks. What do you get from DNA? mRNA. Where does the mRNA go? To the ribosome, where who brings amino acids? tRNA. And then we make polypeptides, and if you put enough polypeptides together, you get proteins.
Where's our RNA coming
Why is she laughing so hard?
Where's our RNA coming in?
Where does RNA come from?
Come in.
Pirates.
Or
Arr.
where's it coming in?
Oh, no, it's RNA. Who can answer that question?
Ribosome.
They make up ribosomes, right? That's what RNA is. Okay? How are we doing? How are we feeling? Feeling good?
What's an enhancer?
What is an enhancer? It's kind of a binding site for transcription factors. Okay? It's pretty much a binding site for transcription factors. All right, let's keep going. The question then is why? Well, there's two parts to this question. Why is it that eukaryotic gene regulation is so much more complicated than prokaryotic gene regulation? Who's got the answer to that? There's two answers, really. Yes, Cole.
It's because eukaryotic is more complicated than prokaryotic.
Yeah. I mean, if you look at a prokaryotic cell and a eukaryotic cells are just a lot more complicated. That's just the way it is. And they're a lot bigger. And they have, they have organelles, and they have a nucleus, and they have chromosomes. So that right there is one answer. What's the second answer?
They're specialized.
They're specialized, right. If you are E. coli, you are E. coli. You're a one-room workshop that makes proteins. That's it. If you are a eukaryotic cell, let's take on a Christmas tree that is still living. What are the different type of cells do you have in a tree? A lot. It's always a safe answer, right? But what kind? Do you have chloroplasts?
Yes.
That's an organelle. Do you have cells with lots of chloroplasts?
Yes.
Maybe we should talk about humans. What type of cells do you have?
Blood cells.
Blood cells, nerve cells.
Muscle.
Lots of different cells, right?
Hair cells.
Here's your DNA. Here's your DNA. And each part codes for a different part of your body. So, what does that mean? That means that there are times when some of them want to be turned on and some of them will be turned off, but we need more in order to get more. Prokaryotes are easy. We don't need this much DNA because it's simple. We're always making the same thing. Does that make sense? So when you talk about specialization, we've already talked about specialization. We talked about differentiation. Do you remember that? Remember all the totiponent cells and the pluripotent cells and all those? We're actually talking about those cells, but now we're looking inside. We're looking at the DNA, and we're seeing what's going on with that, okay? So, again, genes that code for the liver enzymes are not needed for brain cells. Is the DNA located in the brain cell, though? Yes. There is instructions for liver enzymes inside of the brain cell, but it doesn't need to be there. So the brain cell wants to say, hey, we don't need this part, we only need this part, so let's focus on that part. Does that make sense? All right. Good deal. How do eukaryotic cells turn off genes? One way we think is a new type of RNA. We talked about mRNA and rRNA and tRNA. Here's a new one. It's called mini-RNA, right? I feel like we're talking about like iPods or something, you know? We got the shuffle and the mini. No, there's no mini, right? There's a shuffle and then the nano, the touch. There's a mini Mac, though. That's where I'm getting that from. Anyway, the point is that we have found that we have these RNA molecules that are sort of floating around in the nucleus and in the cytoplasm. And there's this thing called a dicer, which I always think of some like being at Takioka, and you know that guy, it's like, right? But the dicer is actually going to sort of loop the RNA around and have it fold upon itself, and then it's going to dice them up into small pieces. Then these guys are going to split and they're going to hook on to other RNA. What they're basically going to do, if you look at this, it says no translation, no protein. What does that mean? This right here is mRNA that's going to be headed towards the what? The ribosome. Very good. But what mini RNA does is it binds to it and prevents it from doing that. And at first when researchers found this, they thought this must be a mistake. This is kind of weird. But then they figured out, no, it's not a mistake. What it's doing is it's preventing certain proteins to be made. Because again, are there times when we want to make this protein but not all of these proteins? Yes. So this is a certain way to do that. It's also been very exciting to know that because they said, wait a minute. What if we could use mini RNA to turn off cancer and to turn off viruses? So that's exciting. There's a lot of promise in that. Obviously, it hasn't happened yet, or you probably would have heard about it on the news, right? Let's talk about how in the world this embryo is going to be turned into an actual organism. And it has to do with different types of genes and different types of transcription factors and repressors. And this is where probably your head's starting to hurt a little bit, right? Maybe? Because we started with something simple, prokaryotes, and that made a lot of sense, I think. The lac operon and lactose and operators and promoters. Then we went to eukaryotes, and it's a little more complicated, but I think I know what you're talking about. And now we're going to talk about how do we use genes like the Hox genes to figure out how we look like we look. Hox genes. Kind of sounds like somebody's sick. Like, Hox. No? Okay. Well.
Yeah.
Anyway, so differentiation is what we're talking about. Specialization is definitely what we're talking about. We're going to start with homotic genes. Homotic genes are like a master set of genes that helps identify and make body parts in the embryo. Because if you look at all the cells in the embryo, it's hard to decide which one's going to become the eye and which one's going to become the foot. And you really don't want to mix those up, right? It's pretty important. You don't want a toe on your face and an eye on your feet, right? It's pretty important. You like that one, Maddie? Oh, boy. So, I say that, and you laugh, but he actually did this. He actually, and I say he, I think his name was Lewis. He actually kind of increased the chance of a mutation happen, and we got a fruit fly's leg growing out of its head. Isn't that cool? That'd kind of be cool, right?
So what do you do when you want to land?
Huh? When he wanted to land? Well, he was able to see exactly where he wanted to land, right? Okay, anyway, moving along. If you look at these genes right here, let me show you what's going on with this, because you can see that there's a purple gene, and then there's a purple, what is that? What is that, the mouth? Well, what is that trying to say?
Purple one's the mouth.
It's trying to say that the gene that encodes for the mouth is that one, first. And then it goes all the way down to the blue one, which is its tail. Okay, we call it its tail, even though it doesn't have a tail. Head to tail. So what we're saying is, very simple fruit fly. Goes in order. And the genes that, we find these certain genes, these Hox genes, also in mice. And guess which one's first? The same one, the pink one, is first. It's the same type of gene. But what do we see? We see repeats of Hox genes instead of this. So what would you expect in humans? Same thing, only lots of repeats, right? And that's what we have found with the Hox genes, okay? It's very, very important for development, differentiation, and again, Hox genes are types of homeobox genes that are side to side from head to toe in order, okay? This is something pretty new and pretty...
Wait, so Hox genes are side by side?
These are Hox genes right here. They're side by side in order from head to tail, okay? Basically, their job is to tell the body how to develop. Is to tell this mass of genes how to become a full body organism. And that's pretty crazy if you think about it. The last thing here is really cool because this shows that sometimes the environment, whether it be temperature or population size, this shows how the environment actually can determine when genes are turned on or turned off. And this is pretty cool because what happens if the tadpole notices that the temperature is too